Hello students, today we are going to discuss about 3-point test cross. Okay, 3-point test cross means when the three genes taken into consideration. When the three genes taken into consideration, then it is known as the three-point test cross. The test cross means when the F1 they cross with the recessive parent. Okay, so that are known as the test cross. Okay, so here the 3 points, as like the 2-point test cross, the 2-point means the dihybrid test cross. So here the 3-point test cross, therefore it is known as the trihybrid ratio. It is known as the trihybrid ratio, trihybrid cross. Okay, so in trihybrid ratio, the 8 progenies are produced. When we perform the trihybrid ratio, 8 progenies are produced in the ratio of 1 is to 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 1. Okay. So 1, 2, 3, 4, 5, 6, 7, 8. So these 8 progenies are produced when we perform the trihybrid ratio or trihybrid test cross. Okay. So when there is when suppose there is a plant that having the three characters we have taken into consideration, so these three genes capital A, capital V, capital C, when we cross with small a, small a, small a, small a, small b, small b, small c, small c. Okay, so it produce capital A, capital V, capital C. It produce small a, small b, and small c. Okay, so when we cross these two, it produces capital A, small a, capital B, small b, capital C, small c. Okay. So this F1, when we again back cross with the recessive parent, so it produces a single type of A, B, B. It produces only three gametes, A, B, and C. So it produces only one type of gamete A, B, C with three genes. Okay. But it produces the 8 different types of gametes. Okay, and therefore this ABC with all of these it produces the 8 different progenies. So here we are going to discuss a problem. Okay, so this problem we have to calculate the distance between the three genes. Okay, so the problem is the maize plant, the maize plant having three characters, having three characters of endosperm, that is colored. So it's having the gene capital C, the colored, then full, the gene capital F, and the starchy capital S. So the maize plant that having the three characters of the endosperm, colored, full, and starchy, crossed with, in the crossed with the plant having the characters, recessive characters, so that is colorless, so small c, then it is shrunken, then shrunken, so small f, and waxy, so small s. Okay, so produce the heterozygous progeny in the F1, and that F1 when crossed with when heterozygous when heterozygous F1 progeny crossed with the recessive parent to perform the 3-point test cross 3-point test cross, the progenies were produced so the number 1 progeny, it is colored full starchy. Second, colorless shrunken waxy. Their number were 2538, 2708. Then the third progeny, colored shrunken waxy. Colored shrunken and waxy, 2116. The fourth, that is colorless full starchy, colorless full and starchy, so they are 113. Then fifth, colored full waxy, colored full and waxy, they were 601. Then sixth progeny, that is colorless shrunken and starchy, colorless shrunken and starchy, so they were 626. Then two more progenies, so we will write here the seventh progeny, so that is color shrunken and starchy. So color shrunken and the starchy, so there were 4. And the eighth progeny, colorless full waxy, that is colorless full and waxy 2. So calculate distance between 3 genes. Okay. So it is the problem. The maize plant having 3 characters of endosperm, that is colored, full and starchy, crossed with the colorless, shrunken and waxy. Okay. So when heterozygous F1 progeny crossed with recessive parent to perform 3-point test cross, the progenies were produced. So that is colored full starchy 2538. Second, colorless shrunken waxy 2708. Third, colored shrunken waxy 116. Colorless full starchy 113. Colored full waxy 601. Colorless shrunken starchy 626. Colorless full waxy 2. And color shrunken starchy 4. Okay. Now we have to calculate the distance between the three genes. Okay, so now solve this problem. Okay, solve the problem. So we know that the formula for crossing is the percent crossing over is equal to non-parental non-parental progenies, or we can say that the recombinant. So number of non-parental recombinants divided by total number of progenies into 100. Okay, so first of all we have to understand how the crossing over takes place here. So these are the two chromosomes, okay, on which this capital C, capital F and this capital S is present. Okay, similarly the small c, small f and the small s is present. Okay, so it is the C for the color, it is for full and it is for starchy, capital F. Small c for colorless, small f for the shrunken and small s for the waxy. So the total, so the progenies produced they were, okay. So first of all, there is no recombination at all. Okay. So due to no recombination, so there is no recombination. Therefore, it produced first, that is colored, full and starchy. So it produced that progeny, number of progenies that were 2538. 538. Okay, 2538. Then this one, that is colorless, shrunken and waxy. So colorless, shrunken and waxy. So the number of them 2708. 2708. Okay. Then there is the first crossover takes place at this place. Okay. So when this crossover takes place, so it produces this capital C, it goes with this small s. So it produces the colored, shrunken and waxy, and fourth one, that is small c, so it is colorless, and this colorless with full and starchy. Colorless, full and starchy. So these progenies were produced, their number was 116, 116, and its number 113. Okay, then suppose the crossover at the second place. Okay, it is the first, it is second and it is third gene. Okay, so first cross, there was a single crossover at 1 and 2 gene. Okay, or in between 1 and 2 gene. Now the second crossover between second and third gene. So here. So the progeny is produced where? The fifth one. That is colored, full and waxy. That is colored, full and waxy. And six, that is colorless, shrunken and starchy. So colorless, shrunken and starchy. Okay. So this progeny is where the colored, full, waxy, the 601. That is 601. Okay. And these two, it is the second 626. Okay. Now the seventh and the eighth progeny. Okay. So now there is a double crossover between these two. That means at this place also and at this place also. There is a double crossover. Okay. And due to that double crossover, what happened? This capital C, it goes with this small f and it again goes with the capital S. And therefore, that is the colored, shrunken and full. Colored, shrunken and full. And they were 4. Okay. And now this small c, capital F and small s. So colorless, he said colorless, so this colorless, then full and then waxy. Colorless, full, this colorless, full and waxy. And there were 2. Okay. So now we have to calculate the percent crossing over between all these. Okay. That means between crossing over between this C and F, then crossing over between this F and S, and the total crossing over between C and S. Okay. So now the formula is percent crossing over is equal to the number of recombinant divided by the total number of progeny into 100. Okay. So first of all, we have to calculate the crossing over distance between C and F. So here the percent crossing over between the C and F. Okay. So if there is we have to calculate the C and F. So we have to calculate only these progenies. Okay. Which were produced due to the C and F crossing over. The crossing over at C and F. Okay. So we will draw here. The total number of progeny 2538 plus 2708. These are the parental plus 116 plus 113 plus 601 plus 626 plus 4 plus 2. Okay. So first of all, this crossing over between C and F. Okay, this C and F. So first crossing over, these two were produced. Okay. So this 116 and 113. So these are total progenies 2, 2 and 9, 229. So 229. divided by total number of progenies. How many total number of progenies there were? So the total number of progenies were 6708. Okay. So here 6708 and into 100. So it produces the result 3.4%. Okay. We know that the 1% means 1 unit, and that unit is 1 centimorgan or 1 1 morgan unit. Okay. Here the 3.4. So the distance between C and F is 3.4. Okay. Now we have to calculate the crossing over progenies between or distance between F and S. Okay. So okay. So F and S. So distance between F and S. So these are these two progenies. These are these number of progenies. Okay. This fifth and sixth, they are produced due to this. Okay. So these are 601 and 626. So the sum of it produced 1227. Okay. So total number of progeny 100 1227 divided by the total number of progeny 6000. So 1227 and total number of progeny how many? 6708. Okay, into 100. So it produces 18.2%. It produces 18.2 or 24. So this distance is 18.2%. Okay. So now the CF is over, FS is over, now C and S. Okay. So the distance between C and S. Okay. So these two progeny, they are produced due to the double crossover and they were 6 only. So 6 divided by 6700, these are total progenies into 100. Okay. So this gives the result 0.0894%. Okay. 0.0894%. Okay. So it is due to the double crossover. It is the result of double crossover. It is not the distance between this F and S. Now we have to calculate the percentage of recombinant, and then we can calculate the distance between C and S. Okay. So we will calculate here. Okay. Okay. So now we have to calculate percent recombination. Okay. Percent recombination. So the percent recombination in first of all is C and F. Okay, in between these two. So we already calculated the distance between that. It is 3.4. Okay, 3.4. It is 3.4 is the result of single crossover. Okay, and now what is the result of double crossover? The double crossover. The result of the double crossover is 0.0894. Okay, 0.0894. But it is between this CF and FS. Okay, so we have to divide it by 2 because it is between this CF as well as FS. So there are the three genes. So this combination is 1 and it is 2. Therefore we have to divide it by the 2. So 3.4 plus this results into 0.0447. Okay, so when we calculate 3.4 plus 3.4 plus 3.4447. Okay, it is the crossover between this, that is 3.4447. So it is the 3.444 sigma. Okay, it is the percent recombination between C and F. Now we will calculate the percent recombination. We will calculate the percent recombination between F and S. Okay. The formula is same. Okay. So what is the? It is now the percent recombination in F and S is equal to, now it is 18.2. Okay, so 18.2 plus, again we have to divide it by 2. Okay, so here 18.2 plus 0.0447. Okay, so it is 18.244 Okay, so the recombination recombination between C and S, this C and S. Okay, so here What is that? 3.4447 plus 18.2447 means it is 21.68. Okay, so the distance between two, it is 21.68 Morgan unit. Okay, got it? See the distance between C and F. It is the distance between this and this. So we have calculated both single crossing over as well as double crossing over, and due to that the recombination, we have calculated how many recombinations there, and according to that we have calculated the distance between CF, FS and then the total distance. So total distance is 21.68. Okay, so it is the chromosome on which here the C, here the F and here the S. Okay, so distance between C and F, it is 3.4447. Distance between F and S, that is 18.2447. Okay, and the distance, the total distance between C to S, that is 21.68 Morgan unit. Okay, so it is the clear conclusion that the number of progenies due to the single crossover and double crossover between the C and F, they were less, and therefore distance between C and F is less, while the recombinants due to the F and S, they produce more in number, and therefore the distance between F and S is more. It has the more distance because more recombination. Okay, and here the less distance than FS because the less recombination. Okay, so in this way we can calculate the distance between the genes. It is known as the gene mapping. It is known as the gene mapping. It is known as the gene mapping. So it is the 3-point test cross. 3-point test cross. Okay. I hope you got the points. Thank you very much.